Next speaker is Harry Feng. Harry Feng is assistant professor at Case Western Reserve University School of Medicine and associate member of Case Comprehensive Cancer Center. How is the author of DSS and NeuCA bioconductor packages. In particular, NeuCA is a new addition to the recently released bioconductor version 3.14. So I would like to turn things over to Harry, please.
Thank you, Kozo, for the nice introduction, and it's my great pleasure to talk here today. Let me share my slides. Okay, you should be able to see my slides right now. So my name is Feng, and I also go by name Harry. Currently, I'm an assistant professor in the Department of Population and Quantitative Health Sciences at Case Western Reserve University. Good afternoon, everyone, from Cleveland, Ohio, United States. So today I'm going to talk about a new work, which is called NeuCA. So it is really a neural network-based methods for exhaustive single cell label assignment tool. Here is my contact information and my lab webpage. So let's get started. So first, single cell RNA-seq. What is single cell RNA-seq? We know that human tissues have very diverse cell types and that diverse cell states. The traditional RNA-seq, or we call them bulk RNA-seq, can only measure the average signals across millions of cells, right? Single cell RNA-seq or scRNA-seq really give us the first data-driven opportunity to study heterogeneous tissues at a single cell resolution. So this is really a nice tool for us to use. There are, of course, many questions and issues involved in single cell RNA-seq data analysis. For example, from data pre-processing, there will be data normalization issues, batch effect correction, imputation, and in data analysis, there is like cell clustering problems, differential expression, cell type identification, so on and so forth. And also there will be realization problems we have to deal with. In today's talk, NeuCA really focused on one aspect of the data analysis, which is a cell type identification. And I'll focus on this throughout this talk. So we know that the single cell RNA-seq data output is anonymous in terms of the cell identity. We have the output, but we don't know which cell belongs to which cell type, and how do we correctly annotate those cells is a big challenge. It is often the key task in single cell RNA-seq data analysis. This is a big issue here. Traditionally, there are many methods out there available currently. They can take a two-step approach. For example, they can do a clustering, like an unsupervised clustering first to cluster cells into different clusters. And then based on the marker gene they have identified, they will label those clusters into a certain category or certain cell type. For example, Surat can do that at C3, T-scan, for example. But this is a relatively laborious procedure because we have to first cluster and then find the marker genes, and next we're going to label those clusters. And it's also a time-consuming procedure. There's also another problem is that the projection from a higher dimension of the single cell RNA-seq data to a lower dimension representation clustering does not guarantee the best projection. And this procedure highly rely on marker gene. This is also another problem. And there are also additional methods available there to do this in one step, like a supervised labeling approach. Methods around this category include SCMap, Cheetah, cell assign, etc. So they can be categorized into three different categories. So for example, they are really marker gene based to do the cell label assignment, or correlation based, or tree structure based. So those are the three different possible choices. But as a supervised learning tool, it's really not a best idea to use that for novel cell type discovery, because what we can predict, it really depends on what I've learned in the training stage. So those are the existing methods summary. So in this work, we're really going to propose a new method to cell annotation, and we call them NeuCA because it is purely based on a neural network-based cell annotation tool. And we try to make this tool exhaustive. So that means we try to annotate all the cells as much as possible so that the cells cannot be assigned to a certain cell type will be minimized or even eliminated. And also we want to try to build a reliable tool because currently we have already collected massive amount of well-studied existing single-cell RNA-seq data. We want to try to leverage on them. And also we want to try to be flexible because we want to adopt different strategies depending on certain features like correlation, so that our methods can be reasonably flexible. So here is really a motivation of our work. If we take a look at this correlation heat map here, I'm showing you the cell types from a PBMC, the peripheral blood mononuclear cells. And we take a look at their cell correlation plot, and what do we find? So we really find two different patterns, like the CD56 NK cells, and for example, among regulatory T cells, they do not have a strong correlation. Okay, they do not have a strong correlation, meaning that they are easily distinguishable. Okay, they can be easily separated without much trouble. But what really brings the problem is those highly correlated cell types. If you take a look at the lower left corner of the heat map, we do have several types of T cells within this category, and they are highly correlated. Highly correlated means it's more challenging and more difficult for us to distinguish them. For example, if you take a look at the regulatory T cell, CD4 T cell helper, and naive T cells, they have strong correlation structure, and this gives us challenges. So what we are thinking is, why don't we use this tree structure and this correlation and adopt different strategies to make a prediction methods? So here I'm taking a training data as an example. So first, we can definitely calculate the correlations of the mean cell type expressions first, because the training cells, training data have been labeled, and we can calculate the correlation. And then depending on the correlation, whether this is a very strong correlation or this is a low correlation, we can adopt different strategies to distinguish those cell types and do the prediction. So this is our, brings us to the details of our proposed methods, NeuCA. So if we first focusing on the top left corner, this is basically the same as the last slides. We have the training data, and the training data are very well labeled. So we can get the heat map and get the correlations about the mean structure. And depending on whether we have a high correlation or not, we can choose from the two different approaches. Okay, so first, let's take a look at the approach on the lower left corner of the this picture, the cyan color panel. So if the cell types are not highly correlated, we can simply use a vanilla style feed-forward neural network to train a classifier and to do the prediction for testing data. That is easy. And if the cell types are highly correlated, meaning this is a very challenging problem, we can adopt the strategies on the right-hand side of this panel, which is shown in the pinky panel here, is that first we will learn this hierarchy tree structure, and then we will simply replace the node of the tree structure with this neural network so that it can help us to do the prediction. As you can see from the top right corner, we have learned this tree structure from this training data set, And then in the next, we will replace each node with a neural network so that to help us identify the and predict the cell type. So this is really the two alternative strategies that NeuCA can take depending on the correlation structure. So here are some technical details for the for NeuCA. I'm not going to talk anything in details, but just leave them here for you to refer to in case you are interested in how the model was built. For example, we have 3 different model sizes, big, medium, small, representing the number of nodes in the neural network and the number of layers in the neural network. I'll just leave them here for now. And we did some simulations to show how NeuCA performs. So the simulation is really based on real PBMC data. We take the 10x PBMC single cell RNA-seq data, and we did 80 Monte Carlo simulations and aggregated them together to show the results. And we also know changes the proportion of the training samples using only 10% of data as the training, all the way to using 80% of the data as the training. So here are some simulation results. It's real data based. So as you can see from the first 3 columns, the first 3 columns are the different model sizes of our proposed method NeuCA, and the remaining columns are the existing ones like SCMap cluster, SCMap cell, cheetah, and also we compare with those unsupervised learning approaches like SURAT and SC3, and we use in panel B here. We use ARI, adjusted random index, to show the difference. And in the first panel A, we simply use the accuracy to represent our results. As you can see from the panel here, our proposed method, which are the first 3 columns, first 3 box plot, shows a higher accuracy on average and a higher ARI on average compared with existing methods. We also conducted additional real data-based simulations by changing the number of training cells we need. And it's really striking for us to see that from the top row of the result, as you can see, we only use 10% of the data as the training data sets. And in the bottom row, panel C and D, we are only using 20% about all the cells as the training data set. We can see that even if we only use 10% as the training data set, we still achieve a high accuracy for 2 situations using NeuCA. And it shows a clear advantage over existing methods by only using a small proportion of the data as the training. And a similar conclusion can be drawn by using 20% of the data as training. Going back to the motivation page, as I showed you earlier, so those several highly correlated T cells are here. And how does our method perform on those highly correlated cell types? Let's take a look on our next page for those highly correlated T cells. So it might be hard to see, but for each panel here, the first bar here is representing the true cell labels, and the second bar here is representing our proposed methods, and so on and so forth. There are additional methods that lays out for each bar. Okay, and each cluster, each panel here representing 1 cell type, and we can see that pretty much all the methods perform reasonably well on the B cells, for example, and also performs very well on the CD56 NK cells. Let's go to the T cells part. We can see that for our method, which is the second bar here, really a close mimic to the first bar, which is the ground truth. So that means our method performed very well in the highly correlated T cells in general. But there are exceptions like naive T, we didn't do very well, but we did a pretty good job on CD4 helpers, naive cytotics T cells. So this shows a picture on the average around multiple rounds of simulations. This also shows a potential advantage of our proposed methods. So let's move on to some real data results. So here is also based on PBMC data, peripheral blood mononuclear cells. Here we are really collecting single cell data from multiple projects and put them together to see if we train on one of them and try to predict the rest of the studies, can we do a good job? And so we're trying to answer this question. So here we took data from different sources and try to use different methods to train and then to predict. And we can see that from the 4 panels here, we're trying alternating the order of the training and testing. And we found that our method is pretty consistent and continually ranks the highest among all the potential methods available in this simulation comparison. And this shows the first bar here is our methods that ranks reasonably high enough. And also here is another way to visualize how methods performs. As you can see, this is a t-SNE visualization, and the top left corner is the true class rings, and our method is on the top right corner. And what's really makes it interesting compared with the existing method is that if you look at the lower row of the panel, there are lots of gray colored dots here. Those gray color actually means they are unassigned cells, like around here and here, as you can see. So for existing methods, they do produce lots of unassigned cell types, meaning the software, the method is not sure which cell types are they. So they assign them as unsure. But in our method, we try very hard to eliminate this unsure problem, so that pretty much all the cells can be assigned to a certain category, and this category is very consistent with the original visualization. And finally, so here is a pancreas data for real data results. So this is a Sankey diagram showing the truth versus the predicted. On the bar on the left showing the truth, and the bar on the right shows the prediction for our results, which is the first row, and also the IC map cells on the second row. We can see that we have a pretty consistent flow chart on Sankey diagram in this panel. But for existing methods like SC-cell, they produce a high number of unassigned cells, which is not ideal for downstream analysis. Okay, and that's pretty much all I have in terms of simulation. And let's move on to the actual softwares. I'm very glad to show this NeuCA package recently released with the release of the bioconductor version on 3.14. And right now it's officially there. And feel free to download it and give it a try. And also we have a higher level wrapper function available to use. It's really easy to use. The usage is simple. Basically, you just provide your training data set, and also you provide the testing data set, and you make a selection of the model size and run it. Within a couple of minutes, it should give you the predicted results. Now, notice that for the training data set, you have to have the labels, right? But for the testings, the labels can be left out as empty, and NeuCA can predict that. Last but not least, it's my great pleasure to introduce this NeuCA web server hosted on our Shiny app by RStudio. So this is a really one-stop place for researchers to use if the researcher don't want to do heavy codings or use the softwares on their laptop or desktop. We are providing this web server to do a single cell label assignment. And in addition to that, we actually collected raw data sets from different places, and we built in some very well-trained classifiers that are ready to use for researchers. For example, if you take a look at the list on the right-hand side, we have collected data from all those places and trained those NeuCA classifiers that are ready to use. If you have a study that lines in with these categories, you can simply upload your data and do the prediction right away within this web page without any coding issues. And I believe right now this list is also expanding. We will have more than 20 well-trained classifiers available right now on NeuCA web server. And feel free to give it a try. And this is a illustration of the graphical user interface. As you can see, or you can really choose from the built-in pre-trained classifiers, and in the drop-down menu, you can select what type of tissues you are working on, and just upload your data and click the button and download the results. So here's the overall summary. This is a one-step supervised learning methods for single-cell label assignment. It's a neural network-based classifier. It's flexible. It means that can adopt different approaches depending on the correlation structure. And it's performed very well, even with a low amount of training data sets. And it has high accuracy. And we have this bioconductor software, as well as this web server for free. So this is the hexagon logo for NeuCA. And last, let me acknowledge my collaborator, Dr. Ziyi Li. She's an assistant professor at MD Anderson Cancer Center at University of Texas. And also Dao Yuduan, he is a second-year PhD student in my lab. And this work is partially supported by intramural fundings from our university. And here's my contact information. With that, I'm wrapping up and happy to take any questions you may have.
Thank you, Harry. I have received some questions for you. One is from Koki Tsuzaki. Is there a global consensus on what is the appropriate resolution for cell types or number of clusters to set?
Yeah, I think that's a very good question. And it's really, from my experience, it's really a back and forth question with the bioinformatician and the collaborator who collect the data. So it's really tricky because we can always zoom in to find a cell type or even find a cell subtype, and we can keep expanding the number of cell types available do that. So I believe we have to have this is a tricky place. We have to make a cutoff decision. For example, if the collaborator believes there are 12 cell types available out there, we can really try to zoom into slightly higher than that and try to find out what each cluster represents, and then find out what each cell type represents. But again, I believe this is really a question we have to answer jointly with the collaborator and biologist. It's really depending on the tissue you are working on and the population you are working on. So that's that will be the best way to determine a number of clusters.
Thank you. And another question from Kokitsuzaki. Does the NeuCA need GPU or GPU? Oh,
okay, yeah. We really called the TensorFlow and the Keras within our program, and currently we can support neural network training without the usage of the GPU. So feel free to give it a try. GPU is not a problem in our case.
Okay. So another question also from Koki. Is it possible to retrain the model with other users' data, for example, transfer learning or something?
Okay, yes. I believe as long as you provide the raw single-cell RNA-seq data as the gene expression count, and provided that as a single-cell experiment object, you can train the model by yourself without using the pre-trained classifiers.
Okay. There is another question. When performing the model against different researchers' datasets to detect common cell types. Have you devised some way to exclude the batch effects?
Oh, a batch effect. Yeah, batch effect is a good issue. I'm afraid my work does not align with that area. But we do have an internal data normalization step integrated in NeuCA. So as long as you provide the raw data count, we have an internal normalization to help slightly alleviate certain batch effect issues. But if you prefer to do batch effect corrections before doing that, I still encourage for you to do so. Thanks.
Okay, it seems I finished all questions, so we already ran out the time. So thank you very much, Hari.
Thank you, Kozo.